Welcome to the basics of neuroscience. Today, we are exploring a deeply important topic, the neurophysiology of depression. We will uncover the biological and chemical realities behind this condition, moving beyond psychology to understand exactly what happens inside the brain. By the end of this lesson, you will see depression not as a personal failing, but as a highly treatable medical condition. Let us dive in. To start, imagine your brain is a massive, bustling city. The neurons are the citizens, constantly communicating with each other using chemical text messages. In a healthy brain, these messages flow rapidly, keeping the city vibrant and energetic. But in a brain experiencing depression, it is as if a heavy, suffocating fog has rolled in. This fog slows down all the messengers, making communication incredibly difficult. The specific chemicals responsible for happiness, motivation, and energy simply do not reach their destinations. This breakdown leaves a person feeling exhausted and disconnected. Understanding this failure is our first step to lifting that fog. Now that we have an intuitive picture, let us look at the formal biology. The most prominent scientific explanation for major depressive disorder is the monoamine hypothesis. This hypothesis suggests that depression is caused by a severe deficiency in specific neurotransmitters called monoamines. There are three main players here. First, serotonin, which regulates your mood and sleep. Second, norepinephrine, which controls alertness and physical energy. And third, dopamine, which is responsible for feelings of reward and motivation. When these chemicals drop below normal levels, the brain simply cannot generate the positive feelings and energy required for daily life. Beyond just a chemical imbalance, modern neuroscience points to structural changes in the brain. This brings us to the neuroplasticity hypothesis. Chronic stress actually reduces the brain's ability to grow new neurons and form new connections, particularly in the hippocampus, which is the center for memory and emotion. Furthermore, depression is strongly linked to a hyperactive hypothalamic pituitary adrenal axis, or HPA axis. When this system is overworked, it floods the brain with toxic, chronically high levels of the stress hormone cortisol. Together, these chemical, structural, and hormonal disruptions create the profound emotional and physical symptoms of depression. Understanding these structural and chemical changes is absolutely critical because it changes how we treat the disease. It transforms a deeply misunderstood psychological struggle into a treatable biological medical condition. For decades, people incorrectly believed depression was just a weakness of character. But by mapping out exact neurotransmitter deficiencies, scientists developed life-saving pharmacological treatments like selective serotonin reuptake inhibitors, or SSRIs. These medications help restore chemical balance and rebuild neural pathways. This biological perspective also emphasizes why therapy and exercise are so important. They physically help rebuild the brain's neural networks, giving millions of people their lives back. To truly understand how treatments like SSRIs work, we can look at the mathematics of the synapse. This formula represents the steady-state concentration of a neurotransmitter in the synaptic cleft. The variable C-syn is the concentration of the chemical available to the brain. In the numerator, R-rel stands for the constant rate at which the presynaptic neuron releases the neurotransmitter. The denominator represents how fast the chemical is cleared away. It consists of K-up, the rate of reuptake by transporter proteins pulling the chemical back, and K-deg, the rate of enzymatic degradation destroying it. Physically, if the reuptake rate decreases, the denominator gets smaller, which forces the overall synaptic concentration to increase, improving the patient's mood. Let us see this formula in action with a concrete example. We have a patient suffering from depression with a serotonin release rate of 120 molecules per millisecond. The natural degradation rate is 2, and the reuptake rate is 8. We need to find the steady-state concentration of serotonin in the synapse before any treatment. We substitute the release rate of 120 into the numerator. In the denominator, we add the reuptake rate of 8 and the degradation rate of 2. Dividing 120 by 10 gives us a final steady-state concentration of 12 molecules per cubic micrometer. This low concentration is simply insufficient to activate the mood-regulating circuits, leading to depressive symptoms. Now, let us model the therapeutic effect of an SSRI medication on this exact same patient. The baseline release rate remains constant at 120, and the degradation rate remains unchanged at 2. However, the medication successfully blocks the transporter proteins, dropping the reuptake rate drastically down to 2. We substitute these new values into our formula. The new denominator is now reduced to 4. Dividing 120 by 4 gives us a new steady-state concentration of 30 molecules per cubic micrometer. Physically, this means the medication allowed serotonin to linger in the synapse much longer, increasing the chemical signal strength by 150% and providing the necessary stimulation to lift the depression. To visualize the math we just calculated, let us look at this diagram of a serotonin synapse. At the top, you see the presynaptic neuron, which is releasing the blue serotonin molecules into the gap known as the synaptic cleft. At the bottom is the receiving neuron, with its postsynaptic receptors waiting for the signal. Notice the structures labeled reuptake transporter on the top neuron. Normally, these act like vacuum cleaners, pulling serotonin out of the cleft. But here, the red crosses represent the SSRI blockade. By physically blocking these transporters, the medication forces the serotonin to stay in the cleft, exactly as our formula predicted, ensuring the receiving neuron gets a strong, consistent signal. We mentioned earlier that depression also involves a hyperactive stress response. Let us look at the mathematical model for how the brain clears stress hormones, like cortisol. This is a standard exponential decay formula. C of T represents the concentration of cortisol remaining after a certain time. C0 is the initial spike of cortisol immediately following a stressful event. The mathematical constant E is raised to the power of negative K times T, where K is the clearance rate constant and T is the elapsed time. Physically, a higher clearance rate K means the body rapidly neutralizes the stress hormone. If K is too low, the hormone lingers, causing chronic anxiety and damage to the brain. Let us apply this decay formula to compare a healthy brain and a depressed brain. First, the healthy patient. They experience an initial cortisol spike of 200 micrograms per deciliter. Their robust clearance rate is 0.5 per hour, and we want to check the levels after 2 hours. We plug the initial concentration of 200, the rate of 0.5, and the time of 2 hours into our equation. The exponent simplifies to -1. Evaluating e to the -1 gives approximately 0.368. Multiplying this by 200 yields a final concentration of 73.6. The healthy brain has rapidly cleared the stress hormone, allowing the body to recover. Now, let us see what happens in the depressed patient with an impaired negative feedback loop. They have the exact same initial cortisol spike of 200 and the same elapsed time of 2 hours. However, their clearance rate is severely reduced to just zero point 0.1. We substitute these values into the exponent, which now simplifies to only negative zero point 0.2. Evaluating e to the negative zero point 0.2 gives a much larger decimal of 0.818. Multiplying by 200 yields a dangerously high final concentration of 163.6 micrograms per deciliter. The math clearly shows the stress hormone lingering far too long, leading to severe neurotoxicity and persistent depressive symptoms. We have covered a lot of ground today, so let us summarize the most important ideas. First, the neurophysiology of depression shows us that this is a complex biological condition, not a personal failing. It involves severe deficiencies in vital neurotransmitters like serotonin, alongside impaired neuroplasticity and a hyperactive stress response system. Second, by using mathematical models, we can clearly see how treatments like SSRIs physically restore these delicate biological balances in the brain. You have now seen the exact mechanisms that empower us to treat depression effectively. This scientific perspective brings hope to millions, proving that their suffering has a clear biological root that can absolutely be healed.